Hello everyone, I am Dr. Vijayalakshmi, Assistant Professor of Zoology at Sri Sharda College for Women, Salem. Welcome to this presentation on methods to culture embryo, techniques and applications in embryo development. Embryo culture is a key process in reproductive biology, helping in assisted reproduction, research and animal breeding. Have you ever wondered how scientists grow embryos outside the body? Well, today we are going to explore different techniques used to culture embryos, understand the science behind them, and discuss their applications in medicine and agriculture. Let's get started. Let's begin by introduction to embryo culture. So, what exactly is embryo culture? It is a process where an embryo is grown outside the mother's body in controlled environment. This technique is very used in IVF, that is, in vitro fertilization, cloning, genetic studies, and livestock breeding programs. The main goal to provide the best possible conditions for the embryo development until it's ready for implantations or further research. Now let's talk about the different methods used to culture embryos. Each technique serves a specific purpose depending on their research or medical applications. Here are some of the main methods. One is in vitro embryo culture. Second one is in vivo embryo culture, and third one is ex vivo culture. So first let's see in vitro embryo culture. It's a growing embryos in lab using a specialized culture media. In vivo embryo culture, it's allowing embryos to develop within host animal before transferring them. And the final one is ex vivo culture. It involves maintaining and studying cells, tissues, or organ outside the living organisms in controlled environment. It allows researchers to analyze biological processes, test drug responses, and studying disease mechanisms while preserving the native tissues architectures. Let's understand in vitro embryo culture. One of the most widely used techniques in reproductive medicine is in vitro embryo culture. It involves retrieving oocytes, that is, eggs, fertilizing them in laboratory conditions, and growing them in nutrient-rich medium until they reach the blastocyst stage. In IVF clinics, these embryos are carefully monitored under microscope to assess their development before implantations. So the following three key aspects are very important. One is controlled lab environment. Second one is required specialized culture media, and final one is commonly used in IVF and genetic research. Unlike in vitro culture, in vivo embryo culture allows embryos to develop inside the living organisms before being transferred. This method is frequently used in animal breeding, where fertilized embryos are placed in surrogate mother. So while it provides a more natural environment, it lacks the precise control of in vitro methods. As we know, culture media play a crucial role in supporting embryo development by providing essential nutrients, energy sources, and growth factors required for cell division and differentiations. These media are carefully formulated to mimic the natural environment of the embryo, ensuring optimal growth conditions. First, we see simple media. So, simple media are designed primarily for the early stage of embryo development. They contain a basic salts and glucose, which serves as a fundamental energy sources. These components help maintain osmotic balance and provide the necessary nutrients for initial cell division and growth. Then complex media, this is also very important. As the embryos progresses to later developmental stages, it's required a more diverse range of nutrients. Complex media includes amino acids, vitamins, and growth factors which support cellular metabolism, protein synthesis, and overall embryonic development. These additional components enhances the embryo ability to grow and development properly. And final one is the sequential media. Since the nutritional requirements of the embryo change over time, sequential media are used in a stepwise manner. different culture media are applied at specific developmental stages to provide the precise nutrients needed at each phases. So this approach ensures that the embryos receives optimal support throughout its growth, improving viability and developmental successes. So using the right type of culture media is essential is assisted reproductive technologies like ART and research applications, as it directly influences the quality and potential of the total development of embryo. Now, let's look at some advanced embryo culture techniques. First, we see whole embryo culture. This method is essential for studying early embryo development outside the body, helping researchers understand congenital defects, genetic modifications, and the effect of drugs on embryos. The organ culture allows scientists to grow whole or partial organs in the laboratory while maintaining their structures and functions. It's mainly used in regenerative medicine, drug testing, and transplantation research. And third one is microdrop cultures. This technique is highly efficient in IVF laboratories. Embryos are placed in tiny droplets of culture media under mineral oil, ensuring a stable pH and moisture level for improved embryo viability. And fourth one is co-culture systems. Ever wondered how scientists mimic real life conditions in the laboratory? The co-culture system does exactly that by growing different cell types together. This technique is crucial for stem cell research, embryo development studies, and even improving IVF success rates. And final one is rotating bottle cultures. Imagine system that's constantly keep tissues in motions for better nutrient absorption. So that's exactly what a rotating bottle culture does. It's widely used in large scale cell cultures, tissue engineering, and vaccine productions. Applications of embryo culture. Today's embryos cultures play a huge role in various fields. Let's take a look at its real world applications. Assisted reproductive technology. It helps couples conceive through IVF technology and genetic research. Scientists use the embryos cultures to study genes and congenital disorders, cloning and stem cell research. It's essential for regenerative medicines, agriculture and animal breeding, used to improve livestock breeds and genetic traits. Let's talk about what influences embryo culture success. It's not as simple as just placing embryos in dish. Several factors will play a crucial role. Maintaining a stable temperature and pH is critical. Any slight fluctuations can affect cell growth. Low oxygen levels around 5% mimic natural conditions and promote better embryo viability. A well-balanced culture medium provides essential nutrients for cell divisions and growth. Contaminations from bacteria or fungi can disrupt embryo's development, making sterility a top priority. Let's talk about challenges and limitations. While embryo culture is revolutionary, it comes with challenges, ethical concerns, success rate variability, environmental sensitivity. And coming to the environmental concerns, the use of human embryos in research raises moral questions. Success rate variability is not all cultured embryos survive or develop correctly. And the final one is the environmental sensitivity. Even minor changes in culture conditions can cause developmental defects. Looking ahead, the future of embryo culture is exciting and full of possibilities. Scientists are working on artificial womb technology for better embryo growth, gene editing advancements to prevent genetic disorders, and automated monitoring systems for improved embryo survival rates. With these innovations, embryo culture will continue to shape the future of medicine and agriculture. To sum it up, embryo culture is a game-changing technology in reproductive biology. From assisting infertile couples to advancing genetic research, its impact is vast. So, by understanding different culture techniques, we can appreciate how science is transforming human health and agriculture. I hope this presentation has been helpful in understanding the methods of embryo culture and their importance. Thank you for your attention, and I look forward to seeing you in the next module. Thank you.